Guys, settle down, please. Today we'll be explaining cells chapter, me and Saif. I'm Ahmed Emam, and this is Saif. I'll be explaining the first part, then he will be explaining the second part, and so on. So, as you guys are all familiar with cell theory, as you all know, cell is the smallest unit in life. All organisms are made from one or more cells, and you know that. For example, we're made from like billions of cells. That's easy. Cells only arise from other cells. That's like mitosis and meiosis. Our cells divide, so we arise from other cells. Meiosis, it's the sex cells. Sperms arise from germ cells. Okay, now we have three main parts in a cell. You have the cytoplasm, the nucleus, and the plasma. And cytoplasm, it's the intercellular fluid. It's inside. They hold the organelle, basically. They are the intercellular one. And they perform a specific cell function, which is like they hold the organelles in their place, and it's called the intracellular fluid. The nucleus, it's basically, as you all know, it holds the DNA. يعني typical DNA تبعنا. Okay? With the plasma membrane, it's the outer boundary. It acts as selectively, like a selective membrane that allows ions or nutrients or anything to pass by, as you guys all know. Okay? أول شيء مين هون قرأ الكتاب؟ بالهوا سوا يعني. Okay. يلا سيف. طيب. First of all, that's the surrounding around the cell, which is the extracellular materials, are the substance contributing to the body mass that are found outside the cell. So first of all, it's the extracellular fluid, which is basically the cell. You see, this is the cell. This is the extracellular fluid. Okay, it might be interstitial fluid, as you guys see here, or it might be the plasma or cerebrospinal fluid. So it's basically any cell that's outside the cell. Any fluid that's outside the cell. So interstitial fluid is fluid in tissue. It basically batters the cell, like بتنظفها. Contains amino acids and sugars, like it contains nutrients that might give nutrition to the cells, basically. Cellular secretions, as you guys look in tissues, you know, serous membrane, صح? The cells of it, they give a lubricating substance عشان تمنع الفريكشن, عشان تتحرك, so you don't have a friction. Okay, so that's basically it. And also the cellular secretion, they produce gastric fluid, gastric acids that help in digesting our food. Okay, so just know these two things in the exam. Let's say, for example, what's two examples of cellular secretion? It will be gastric fluid or gastric acid or intestinal acid, or lubricants here, serous, mucus, and saliva. Okay? Extracellular matrix is the most abundant extracellular material, and it's very known in connective tissue, as you all know. Okay? And it's a jelly-like substance, and it's composed mostly of protein and polysaccharides. And it's like the cell glue, and as I said, it's abundant in connective tissue, and it can range from soft to rock. So it can be either soft or rock, and it also has like a bit of viscous material. Okay, now we have the plasma membrane. It has a lot of functions. First of all, it's a physical barrier. So it separates the interstitial fluid, the fluid inside the cell, which is the cytoplasm, okay, from the extracellular fluid, the one that I just explained right now. And we have the mechanical barrier against toxins and harmful bacteria. For example, if there is a bacteria coming, attacking our plasma membrane, it will protect it. Like our plasma membrane has function of protecting our cells. Okay? And cell recognition, which is by carbohydrates. For example, our immune cells would know that's our cells. Because we have carbohydrates outside, that would know, oh, that's our cells, not a foreign cell. It basically has a receptor, the carbohydrate. It gives the immune system or the immune cells information that, oh, that cell is not foreign, it's from our body. Okay? And now we have selective permeability. It determines what goes and what goes inside and what goes outside. It's basically the ion channels that we explain in a bit. And communication, it allows plasma membrane proteins interact with specific chemical messengers and they message into the cell interior. Basically, it's just to function the cell. That's all. Now, انتوا عندكم البلازما membrane مصنوعة من 3 أشياء تعرفوهم: lipids, proteins, and carbohydrates. Cholesterol, yes. And lipids, cholesterol is part of the lipids, basically. Okay, so first of all, as you guys know, the plasma membrane is a fluid mosaic model. It's called the fluid mosaic model. It's a double layer, as you guys can see here. So you have here the hydrophilic head and the hydrophobic tail. the hydrophilic head they lie on both the inner and the outer يعني من برا ومن جوا. Okay. And it's polar and charged. Polar معناها it can react with water. Polar يعني it has a charge يعني it can react. Okay, and it can interact with water. That's why we call it what, hydrophilic. Okay. Now we have the hydrophobic is the non-polar tails basically, and it's hydrophobic because it avoids like interacting with water, and it only allows lipid soluble stuff to go through the phospholipid bilayer. And we also explain it shortly now. But it's mostly it only allows lipid soluble stuff to pass through, but water soluble stuff no. That's why you call it hydrophobic. بتخاف من المي ما بدها إياها. Okay. And here also we have the cholesterol. It's embedded inside the phospholipid basically, and it helps in maintaining the fluidity of the membrane. لأنه last year بالspring exam جابوا إنه what's the function what stabilizes the fluidity of the plasma membrane. Cholesterol. ليش؟ لأنه when you have يعني cholesterol قليل there will be more fluid. لما يكون عندك cholesterol عالي there will be less fluid, so it maintains the fluidity of it. Also phospholipid could be right لانه it doesn't allow like water to enter, بس انه حسبوا الاجابه يعني they both are correct considered phospholipids and cholesterol. Okay. Cholesterol has two regions, polar and non-polar. The polar region is basically the hydrophilic one اللي هي OH. You guys took it in biochem, you know it. Okay, it's the OH اللي هي it can react with water and it can react with anything. The non-polar it's the non-charged one. So مثلا ممكن جابوا لكم امتحان لعبوا بالسؤال ما يحكوا لكم polar، يحكي لكم انه مثلا it's charged. Non-polar is ionized. Polar is ionized, so like it's charged. Non-polar is not charged, so it will not react to anything. Also, as I said, stiffens the membrane. Also, further decreases water solubility of the membrane. So cholesterol doesn't allow like water soluble stuff to pass by. That's it. Yeah. Now proteins. That's the most important component in the membrane after the lipids. It has like functions. First of all, it's a marker. Okay, it tells who is like what's the cell. Is that a neuron? Is that a muscle cell? Basically. Okay, and you have attachments. Basically, also we talk it in the cell junctions in a bit. Attachments, يعني إنه cells they attract each other or like get attached to each other by these proteins, basically. Okay, transport اللي هي they transport like O2 يعني هي الانتجرال بروتينز هي البروتين channels. طيب who could tell me شو كمان انتوا شايفينه من هون خلص never mind. شو it could be also enzymes where they catalyze chemical reactions, and also could be receptors. For example, بيجينا message for example from like a neurotransmitter or something, هم بيكونوا like a receptor وهم they receive these messages. هم البروتينز هذول اللي على الممبرين. Okay, and their function is determine what the function of the membrane can perform. It can also, as I said here, transport, communication, act as a receptor, and joining cells to each other to the extracellular matrix. Proteins with different shapes have different functions. طيب, who could tell me like what's integral protein in general يعني? Okay, more.
One that passes through both hydrophobic and hydrophilic.
Okay, can it be like embedded on only one side of the membrane?
Yes. Yes.
No, it can be as you it can be يعني مثلا سيف قدم شوي السلايدات. كمان. See you guys, ممكن يكون عندكم integral protein يعني بس لهاي بس مو ترانس ممبرين مو كلها، ممكن يكون عندكم واحدة، فممكن يجيبوا لكم في الامتحان يحكي لكم it's not embedded fully, so it could also be integral protein. Okay, so be careful انه it's transmembrane, most of the time it's transmembrane, بس be careful انه ممكن تكون انه هي بس only on one side of the phospholipid. ارجع للسلايد. طيب now we have the integral proteins. They act as they're mostly helpful for receptors with transport. Okay, now firmly inserted into the lipid bilayer as he said. اعطيهم صح، اعطيهم شوكولاته معتز، اعطيهم. By the way, I'll start asking randomly يعني حاجه اقرب المايك عندك وانت وحظك. It's firmly inserted into the lipid bilayer. Okay, and some only show from one face of the membrane as I said. يعني مو كلهم بيكونوا جوا الممبرين، كلهم بس بيكونوا ممكن on the like hydrophilic part or the hydrophobic part. Okay. Also, all integral proteins have hydrophobic and hydrophilic regions, as I said, allow interaction with polar and non-polar sides of the membrane, as well as water and outside of the cell. Also, transmembrane proteins عندهم like basically two functions involved in transport, as I said, اللي هي form channels and pores. For example, هي ال NA, you guys know them, NA channels, calcium channels. هذول الاشياء اللي بتدخل وتطلع برا الممبرين هم اصلا they are the main type of protein that helps in this transport. Okay. Also في عندكم carriers that we'll also talk about in the fusion. انتوا عندكم two types, carrier and channel. في عندكم channel والcarrier. الcarrier اللي هي زي it changes the shape and we'll talk about it shortly. Okay, it can also act as enzymes. What do you guys know about enzymes? أنا بدي أسأل واحد إن شاء الله. Exactly, they speed up the reaction. Exactly. So they could, for example, let's حكوا لكم مثلا إنه a person بالامتحان حكوا لكم مثلا a person had an issue in catalyzing the reaction, or for example, it's not speeding up. What type of protein could be damaged? No, what type of protein? Integral protein. And integral protein يعني هي it acts as an enzyme or as a receptor. Okay. فيعني مثلا حكوا لكم الانزيم خرب, what type of protein could contribute to it? Integral. Okay. مين؟ معتز. Now you guys have peripheral proteins, which is basically embedded into the like full integral protein. يعني مثلا يعني سيف لو سمحت برضه ترجع. بعدين معك. For example, as you guys see, that's integral protein, and the peripheral هي اللي بتكون من برا. But only surrounds it يا اما يعني the outer layer, مو من جوا الفوسفوليبيد. يعني you will never find like a peripheral protein inside, embedded inside. It will just be on the surface, basically. Okay. سيف ارجع. Okay. It's not embedded in the lipid bilayer, attached loosely to integral protein, as I said. Have a hydrophobic region that anchors then into the membrane from like a cytoplasmic side, as I said, from either inside or outside. It also acts as enzymes or motor proteins, or also they link cells together. يعني الموست شو اسمه الموست function to it, as you guys know, لانه يعني هم الاثنين متشابهين, بس انه they link cells together is the peripheral proteins. ليش؟ because هم they can be outside this, يعني outside على الاكسترا فلويد, فهم يكونوا برا, فهم يشبكوا السيلز مع بعض. You know what I mean? سيف؟ هون carbohydrates is the most simple one. بس اللي لازم تعرفوه, when the carbohydrate is embedded into the lipid bilayer, it's called glycolipid. Embedded into the protein, يعني شابكة بالبروتين, as you guys can see here, okay, it's glycoprotein. As simple as that. إذا شابكة بالبروتين glycoprotein, شابكة بالليبid glycolipid. Okay, هم بس you guys use them to like شو اسمه recognize your immune cells can recognize you, as I explained shortly. Okay, انه who is who during development, so cells can sort themselves out. يعني عشان ما تيجي الاميون سيستم وتفكرني انا فورا, okay, وتذبحني انا وتاكلني انا. Okay. They are also found on the outer surface of the membrane like sugar. They are like sugar coated basically, which helps the immune system recognize it. The extracellular surface, but not the intercellular surface of the membrane, is decorated with short branching carbohydrates. It's this one. Okay, and it's also like إذا حكوا لكم مثلا the type of material, إذا حكوا لكم مثلا what type of plasma membrane components has a fuzzy sticky carbohydrate rich area, it's the glycolipids and the glycoproteins, because they are fuzzy and sticky and viscous basically, زي الglue. Here's just basically a small summary. هون هذا البارت في like too many people غلطوا فيه. Now I will explain a question that a lot of people might get mixed in. أول إشي, أنتوا you guys know the cell functions. You have 3 of them. Okay, cell junctions. You have 3. Okay, you have the tight junction, which is like the location of it is what epithelial cells lining the digestive tract and enzymes and microorganisms of the intestine. Basically, if they told you anything related to digestive system or intestines, it's mostly by tight junctions. Like they are like abundantly found there. What's the function of tight junctions? Who could tell me? Exactly. معتذر. طيب. اوكي let's say كمان مش حيجيبوا لكم السؤال straight forward, ممكن يحكوا لكم it's impermeable junctions. اوكي يعني tight junctions, another name is impermeable junctions. ليش؟ it doesn't allow fluids to leak from. For example, ليش هم اصلا مهمين in the digestive tract؟ لانه احنا we have gastric fluids, we have blood. ف if there is no tight junctions, الدم حيسرب, like it will leak out, and بعدين حيصير عندك like edema and like a lot of complications basically. انه your stomach يعني مش حتخزن الاكل, it won't be storing food basically. Okay, and it forms continuous seals around the cell and prevent molecules from passing between cells. Okay, and it's known what in epithelial cells lining the digestive tract and the intestine. Also, it might be a bit leaky. It's normal. It's a bit leaky though, not too leaky عشان بنتوفى بعديها. So it's impermeable junctions. So time junctions are leaky and might allow certain ions to pass. يعني بتخلي ions معينة to leak to the cell. Okay. Desmosomes. Desmosome basically is just قوة, like it resists the tension. So basically, if the cell is going through like a high activity, desmosomes is the one responsible for responding to these tension. Okay. So on the cytoplasmic face of each plasma membrane is a bottle-like thickening called plaque, which is basically, as you can see here, it's this one. هاي البارت بيطلع منها intermediate filaments. Okay. This is type of cytoskeleton. Okay. هاي intermediate filaments بتشبك السيلز they adhere them together عشان يصير الدسموزوم يعرف يمسك حاله منيح. You know what I mean? انه they adhere the cells يعني as you can see here هيها شوفوا هاي intermediate filaments بتشبك مع هاي السيل بتشبك مع هاي السيل بتشبك مع هاي السيل. Okay. And what connects the two plaques together is the cadherins. For example, let's حكوا لكم بالامتحان what's the most abundant protein in desmosomes? There is two proteins actually. Which one is like for the linker one that links? Cadherins or keratin? Cadherins. Keratin is the intermediate filaments which resist the tension. فلسه حكى لكم بالامتحان desmosomes عندهم protein that links the cells together. Which one is it? مين حكى keratin? Exactly, cadherins. Intermediate filament is keratin. Basically, it's just for protection. Okay. Also, we have keratin here, by the way, in our hair and our skin. That's also like the outside layer of our body. We took it with mu'taz and tissues, keratinized and non-keratinized. Okay, and it's also anchoring junctions, as I said. بتمنع إنه cells يتفرعوا عن بعض or tear apart. It's basically just resisting tension. Bind adjacent cell together like Velcro, اللي هو basically like this. تلزقيها وتشيليها. Help keep cells from tearing apart, as I said. Okay. And where it's mostly found, it's in the skin and heart muscle. يعني راح أقول لكم cardiac muscle and skin. Okay. So مثلا راح أقول لكم it's the most abundant in skin and heart muscle. I will come to a really important point after the gap junctions. Gap junctions basically it's for communication. وهم بيستعملوا أي بروتين للcommunication connexons. Connexons عبارة هي البروتين channel that allows the cell to interact and communicate with each other. Okay, as I said here, allows ions and small molecules to pass from one cell to another cell. It's basically just cell to another cell, ions, nutrients, anything. It's just two cells interacting and communicating. Particularly important in heart cell and embryonic cells, اللي هي heart and smooth muscles. Okay, وهي الكونكشنز هي البروتين المهمة إلها. طيب هسه هحكي لكم شغلة. desmosomes موجودة بالهارت muscle والجاب junctions برضه موجودة بالهارت muscle. How will I differentiate between them?
Last junction, upward transfer of ion مثلا او الدسموسوم resist.
Exactly. يعني مثلا let's say جاب لك بالامتحان a 45-year-old patient blah 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 ضل يلعب ويابينج مدري شو. Okay. بعديها he told you he had a problem and the cells like the cell tore apart and it's not functional anymore وفيها like it's basically not healthy enough to resist the tension. So and it's the heart muscle. Which type is it desmosomes or gap junction or tight junctions? Desmosomes, exactly, because resisting tension. بس إذا حكى لك إنه heart وإنه doesn't allow to communicate, it's gap junctions. واللي حيغلط فيه أنا حضربه إذا أجت في الامتحان. Okay. اللي بعده. Okay. Which of the following supports the skin against mechanical stress? لا اللي وراكي. جربي حظك عادي اهبدي كانك في الامتحان. Oh wait say anything. guess from what I explained. لا it's B. I just told you guys mechanical stress is for desmosomes. Desmosome they شو اسمه they resist the tension. Okay. فالكيراتين زي ما انا حكيت لكم كيراتين اللي هي intermediate filaments اللي هي تمسك السيلز مع بعض. الكادهرنز they are just انه شو اسمه they are linked proteins between the membrane. Okay, be careful. Also sorry على فكرة عادي it's okay don't worry about it عادي. Yeah, I was also in this place. It's okay. Let's focus. Okay, now you have the passive transport. Passive transport, it's basically the transport of ions without the use of energy. Basically, it's just like going from uphill to downhill. انتوا for example when you go into downhill مش صح ممكن تسيبوا حالكم وانتوا تنزلوا لحالكم صح؟ That's basically the passive transport. You don't need any energy or any force. Okay, so as you can see here, a substance cross membrane without any energy input. And we also have the active transport. It's from low concentration to high concentration. Low concentration يعني you need energy. ليش؟ لأنكم بتطلعوا uphill. Like بتروحوا من شيء قليل لشيء كبير. So you need energy. It's as if a fight. Like a lot small amount of people going to beat a lot like big group. So هياخدوا معاهم energy. لازم يكون معاهم energy عشان يروحوا يضربوهم basically. Just take it in this sense. Okay. Now, passive transport have three types. I will explain the simple diffusion and facilitated diffusion. Saif will continue with osmosis. The simple diffusion, we'll talk about it. It's just like I'm telling you the passive transport type basically. It's osmosis, facilitated diffusion, and simple diffusion. And the facilitated diffusion, you guys have carrier mediated and channel mediated. Okay, so guys, the rate of diffusion is influenced by, like it could be inhibited or regulated by. Yes, okay, sorry. The magnitude of the concentration gradient means, for example, if you have a high concentration difference, basically, for example, let's say you have low concentration in this area and high concentration in this area. Ions will pass from this high concentration to the low concentration. Yani, stuff will pass like this from high to low, which doesn't need energy at all. Okay, that might cause diffusion to happen. فإذا عندكم difference in, if you guys have difference in concentration gradient, it will diffusion will occur. The temperature of the solution, the rate of diffusion is faster in higher temperature. So let's say, and for example, a person is like in a higher room temperature, or our body has a high temperature. Okay, basically also the diffusion is also faster when the temperature is higher. When it's lower, the diffusion also lower. It's basically proportional. The size of the diffusing molecule, when it's larger, it cannot pass easily. When it's large, when it's small, it can pass easily. Okay, and the viscosity of the solvent. Also, when it's too viscous, it doesn't pass easily. But if it's like water or something that's not viscous, it can pass easily. Also, it depends on the lipid solubility. For example, if the substance is lipid soluble, so it will diffuse more easily. And if it melts or like inside the plasma membrane, it can like pass easily. Yeah. Okay, now diffusion is the movement of molecules or ions from area where it has high concentration to low concentration, as I just explained it, and it's also passive. Okay, and you guys have two types. Simple diffusion. Simple diffusion is basically molecules that can pass through the membrane easily without need of any channels. يعني سيف ارجع لورا. كمان. ارجع هاي. Guys, here simple diffusion, they just pass easily from here. In this area basically, if you guys also don't see here, if they just pass through the phospholipid bilayer easily, ما بتحتاج أي integral protein or channels because they are lipid soluble. Examples of these are like CO2 and O2. Let's say like they said in the exam, which type of molecule can enter easily. If they said oxygen or carbon dioxide, immediately put it immediately because O2 and CO2, they are the only ones that are lipid soluble and can pass through. Okay, its substance directly diffused through the lipid bilayer. Small nonpolar molecules that are lipid soluble like O2, CO2, and also steroid hormones. You guys know the hormone steroid. It's lipid soluble. So steroid hormones can also pass. Let's say حكى لكم thyroid hormone كذا or like blah 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 and keeps and just telling you can it pass easily through the plasma membrane? Yes, it can pass because it's lipid soluble. Okay, and the criteria for it is basically the size, like which regulated, which is basically the size and lipid solubility. So, for example, if it's lipid soluble, as I said, it goes faster. If it's larger, it won't pass. It needs to go smaller to pass easily. And also, water is polar, but it can diffuse through channels, which are called aquaporins, which usually is related more to like osmosis. Now we have the facilitated diffusion, and you guys have two types: carrier or channel. Channel is the transmembrane proteins, اللي هم like integral, as I explained. Okay. Transport H2O and ions through aqueous channels. Also, they are selective due to pore size and the charges of the amino acid. For example, they might be selective, like they might choose the charge, whether it's positive or negative, or they can also select it based on its size. Okay? And you have two types of them, leakage and gated. Leakage, it's always opened. All ions pass through هيك 24/7. They don't close. Gated, it's like a gate. بتسكر وتفتح. Leakage is always leaks, it's always open. Okay, but gated, it's like a door that's closed, and you open it and close it by stimuluses, like from the nervous system or any type of like stimulus. Okay, leakage are always open and allow ions or H2O to move according to the concentration gradient. Also facilitated diffusion is just the used channels, but they also move from high concentration to low concentration. Okay, and they don't need energy. The gated channels, they are controlled, open or closed by electrical or chemical signals, which is basically stimulus. Okay, and they can be inhibited and show saturation and specific. Okay, guys. Also, one important thing when it comes to facilitated diffusion, two things that regulate it is the specificity and the saturation. For example, let's say, glucose want to pass by. Okay, like want to go through the membrane. If all the carriers or all the channels are saturated, can we make glucose pass through? No, because saturated means like they are full. Okay, so saturated means there is no space for them. It's like basically a car, like a taxi. If there is no taxi for the glucose to go in, they cannot pass the membrane basically. Okay, and specificity. For example, if you have like sodium channel, can you make glucose? If you have, for example, calcium channel, can you make glucose pass by? No, because it's only specific for calcium channels. But if it was specific for glucose channel, it can pass by easily. Okay. And as I said, channels can be inhibited, show saturation, and specific substances are also here, like they are. They help in the glucose is like unidirectional. It only goes outside the cell, and H2 and O2 glucose must be transported prospectively because they are vital and they need energy. And now Saif will go on. Do you guys have any questions about this part?
ان شاء الله ما اخبص. هذا بخبص عادي صح. هذا شيء. يلا. بسم الله الرحمن الرحيم. Next. I'll be explaining to you osmosis. We'll start with osmosis. Hello, my name is Saif. Okay, I think most of you know about me. مين قال لا؟ طلع عليك فضائح. What is osmosis? Simply, osmosis is the diffusion or the movement of fluid in through a selective membrane. What are aquaporins? They are like channels that allow only the movement of water. Channels for water. We have channels for sodium, for potassium. We have channels for sodium, for potassium. Those are channels for the movement of water. And what is osmolarity? It is the concentration of a solute in a solvent. So it's the concentration. Next. ركز. ما يكتب؟ يا الله كنت بدي ارسم. مو مهمة يا اخي. سوي اكزت اسمح لي. لازم ارسم. مش عشان اقلد حد بس يعني. من هون. So let's start, okay? So we talked about osmolarity. It is what? The diffusion of water through a selectively permeable membrane. So let's say this is a membrane that only allows the movement of water, okay? So we have equal amounts of water on both sides. You see? Equal. صح or no? And this only allows the movement of what? Water. طيب. Here we have 10 sodium atoms, whatever. 10 moles of sodium. And here we have, for example, 30. Okay, so let's write 30 and here 10. Make sense? Can anyone tell me, if anyone studied already, where do you think water will move? This only allows the movement of what? أبو شرط أسود. From where to where? From left to right. أعطي شيء. Why does this happen? Because the water likes to be يعني هيك حشري. You know, he likes to be between people. Okay? So wherever it sees a gathering of solutes, it wants to be there. So it moves towards the higher concentration of solutes. Okay? So with this example, this explains osmosis. Osmolarity. Osmosis. Okay? So let's try to apply it on another idea. Let's say here we have a bucket, okay, full of water, and here we have this cube of sugar. What do you think will happen to this? Anyone? It will dissolve, صح? It will try to fill this area, all of it, completely. At the end, it will look like something like this. This is the idea of diffusion. Solutes and water try to be like in equal amounts on both sides. So if this only allows the movement of water, what will happen? It will look like this. Because here we have 30, so it will be like this taller, tall P and tall, so I can draw. So it will be like this. Here will be less water, and here will be 30. All of this. Make sense? Any questions? Easy, صح? So let's go to the next part. I'm supposed to open PowerPoint, but I'll not open PowerPoint every time, so I'll post it for myself. We'll talk about the osmotic imbalances, okay? So this talks about osmosis, the movement of water. But let's apply it to cells. For cells, it has a different name. It's called tonicity, which is what we have two things. We have the bucket, okay? This bucket, and we have this cell, okay? Now this cell is a semi-permeable, it has a semi-permeable membrane. And it has what? I said the word. What's the word? Raise hand. صح, aquaporins. This aquaporins allows the movement of only water. صح ولا لا? So let's assume that here we have a lot of solutes. And here we only have like 2 or 3. Where do you think the water will move? From okay, someone hand. صح والله خلصت كلمة. From inside the cell to outside. That means that this will what? Will shrivel. So it will look like something like this. Something like مطعوج الله يساعده. Okay? So, but it will have, so this is called hypertonic. This is what? Hypertonic. عشان أكتبها كاملة يعني. Let's make another example. Here we have a concentration of, let's say, this is 10 liters. Okay, and inside the cell, okay, شوية math يعني, 10 liter with 100. Okay? Inside the cell is 1 liter. Let's say كيف السل فيها اللتر. Okay? And 10 solutes. Where will the water move? Nothing. صح. Why? Because they are equal. This, we call it what? Isotonic. Which means that the inside of the cell and the outside of the cell are both equal. Easy? Make sense? They can bring something like they can bring something like this, where the doctor will give you an example where he says inside the cell there is this amount and the ratio from this to this is like this. So this is a ratio. Even though its numbers are different, but it's the same ratio. So water will move equally. Okay? So this is isotonic. Easy? Any questions? Okay, another example. مين؟ تفضل.
في لتر في السل وبره 10 واسفل.
كيف يعني؟ 10 ما هو؟ ما هو the ratio of this with the ratio of this. So when you divide this over this, it will be the same ratio as this over this. So you need to take the ratio or concentration. صح. It is a ratio that you need to calculate. يعني. Make sense? So if you do 110 over 100, it will be the same as 1 over 10. Easy? Any other questions? خلاص ما حد يسأل. And now for the last case we have here, let's say example, here there is a lot. Okay, he is, dope. He ate a lot. He has a lot of sodium. And here there is only three dots. What will happen? The water will go inside. لازم يشرب مي يعني. So he will be very big and he will explode. مسكين. هاي يعني حرفيا he exploded, and this can be a question. This is what? Hypotonic. So what do we have? Hypotonic. We have hypertonic when the water moves out of the cell. We have isotonic when the movement is equal, and we have hypotonic when it moves inside the cell. Make sense? So let's عندنا أسئلة؟ عندنا أسئلة جاهزة ولا لا؟ بس كله مش مهم. خلاص اصعب. I'll make for you a question, okay? Someone has been eating a lot of what do you call it? كنسل خلاص كنسل. يعني خرف لهم سؤال وما يشتغل. next part شنو هي؟ هاي تنفع. خلاص questions time. سوي شغل. خلاص ما عليك. input. المفروض تنحط عندك بس يلا. ليش مش شغال؟ خراب.
نسوي كيف بيكاسو.
ما هو يا اخي هي هيك تنشرح يا اخي. انا على اللابتوب اشتغلت. ايه يلا سكر عشان ما حد يغش. لا كوي 6 كبرها. يلا a few questions to see if you are awake يعني. The movement of CO2 through the cell is because of what?
Raise your hand.
جاوبت يا اخي. انت ما جاوبت. جيب بس. صحيح فوسفوليبيدز.
يلا.
Next question. Which of the following mechanisms does not follow the saturability and specificity? A. Simple diffusion. B. Ion pumps. C. Carriers or D. Channels. You at the end هذاك blue shirt. B. سوي B ولا لا؟ غلط.
دقيقة. Why did you say B?
صح والله الواي بي.
Why did you say B? Does not follow specificity and
Now we set saturability and specificity. Saturability is when you have like 10 channels.
دقيقة. الأيون بامب. What?
قرب علي عشان المايك هذا.
Okay. الأيون بامب is what is it? Is it facilitated diffusion or too small?
Or simple.
No, it's not active transport. No, I told you it's passive transport. Okay, wait. صح. Why did you choose A? لا, why did you choose A? It's easy. Why? Exactly. It passes easily through the membrane. Okay, that's why it's A. It's simple diffusion. أعطي effort for cheating.
Next question.
Yeah, it's simple diffusion.
Despite
I want to explain something to him fast. Abdullah, ion pump is قلت لك هي the pass of calcium, sodium, so it's facilitated diffusion. It's need channels. وأنا شرحت two stuff that regulates them or inhibit them basically. Okay, it's the saturability and specificity. Okay, so if it's for example you have an ion pump specific for calcium, you can't use it for sodium. And if all the ion pumps for calcium is full, you can't make calcium pass through the membrane. Okay, so be careful. And it's not active transport; it's basic. It's passive. Okay.
Next question. Despite adding more sodium in the extracellular fluid, sodium enters the cell in the same velocity. Which of the following explains this phenomenon? يلا.
Saturability. Why did you
صح. Why not specificity? صحيح.
اوكي وحتى لك معتز اعطيها.
Despite adding sulfate in the extracellular space, it does not enter the cell. Though there are protein carriers. Which of the following explains this phenomenon? هذاك شكله مش فاهم يعني بدي اياه.
يلا اوكي. Why did you choose specificity? That's for another thing. Exactly.
عطوه. خلصت الاسئله يا اخي. وفي اسئله اكثر؟ شنو؟ ثاني سؤال ارجع. هذا؟ اوكي يعني اوكي. So you have, imagine with me, okay? You have 10 doors. Or like for example these two doors. Okay? Now we are here. يعني let's say يعني for example even though we're not 200, imagine 200. Can all of us 200 students go out at the same time? That's what saturability. The doors reach the limit. But let's say we use these two doors as well. we increased the saturability. يعني we added more doors, so it's easier for them to leave. Make sense?
So basically, you have 100 people, and you have like a channel. Okay, when these channels they are all full, every channel can take 10. You have 100 people, and you have 10 channels. Okay, and every channel can only take 10 people. So how many channels you need? 10. وكل واحدة what 10. So and how many watts do you have outside? Like outside 100, صح? So 10, 10, 10, 10, 10, 10, 10, 10. 100 will be full. It will be like fully saturated. It will be full. So if they are all full, can you pass? Can you pass through the channel? No, you don't have a channel to pass through. That's why you need saturability. You need like more channels so it can pass through. Basically, that's it. You know what I mean? فهمت علي؟ عادي. I'll repeat.
Like that, NA plus is very saturated outside.
No, the saturability refers to the channel itself, not to the sodium. So when you talk about saturability, you talk about the door.
Saturability of that. أنا مش حكيت لك facility. You have channel protein and carrier protein. You have channel protein and carrier protein. If the channel is like saturated, يعني it's full, no, like the doors is closed, خلاص, you can't pass through. It's like no other ion can pass through. Then it's saturated. Like you need more channels to pass, basically. That's it. It's not for the sodium itself. It's for the channel. They are saturated. They are full. شغالين. You get it? You get what I mean? عادي. I'll repeat again.
So now because it's saturated, even though it's leaving, it's like something about.
What goes
مش عارف مش فاهم.
بدك ترسم؟ أو الحين ترسم على الرسم؟
امسك هاي.
تشرحها ولا نشرح؟
اشرحها. So معتصم, look. Guys, settle down, please. Wait, I'm gonna دقيقة. شوف, you have this channel, okay? And high, وهاي channel, okay? وهاي channel. Basically, you have these channels, تمام? Every channel, let's say, can take 5 ions, 5 Na. هون 5 Na, هون 5 Na, وهون 5 Na. Okay? Every channel can take 5 Na, صح؟ طيب. Is it one more Na? Can it pass through? Every channel can take only 5 Na. Can this Na pass? حيصيروا 6 Nas. Can it pass? Exactly. It's saturated. خلاص. You cannot make it pass through. Same for the الشبعانة. كلهم خلاص. There is no enough space for it to enter. You know what I mean? No, it's like that way.
Saturability depends on the cell. Like a certain amount enters the cell.
No, that's not saturability. That's the difference in concentration. I'm talking, no. Saturability is for the channels, not for the cell. The cell, it's the concentration difference. Be careful, don't mix up between them. Concentration difference is from here, like, for example, let's see, you have 50 NAs, and for example, here, you have like 30. So,
Depends on the wording of the question.
yeah, exactly. But sometimes these type of questions, they are not straightforward in the exam. They want to see how you think. They are telling you saturability and specificity. So these two are mostly used for what? The channels.
يعني he will not tell you the cell is saturable. He will tell you is the channel.
يعني السيل يكون saturated والسوي الجن. يعني مثلا قال لك after adding a certain ion, after some point the ions stop entering the cell.
Exactly.
The cell is saturated.
Yeah, it's saturated. Exactly.
Even if you added more channel proteins.
No, if you added it, it will not be saturated. الساتوريشن لها علاقة بالchannel. يعني when you لا مو بالconcentration. Okay, be careful.
Saturability, when they're talking about it, they're referring to the rate of diffusion. So how fast my molecules are going to travel. So if I have 5 channels and 5 molecules, the rate of diffusion, let's say, is 1. All of them are happening at the same time. If I increase the number of molecules to diffuse, my rate will not increase. But if I increase the amount of channels, my rate will increase. Why? Because the cell can take more at the same time.
When does the cells stop?
If you're talking about saturated or not saturated, that's going to, you're talking about equilibrium. So basically, you're going to have a lot here and a little bit here. The lot is going to come to the little bit until they're the same. Then it will
channel more concentration difference.
This is all referring to passive diffusion. So we're referring to passive or facilitated, so there's no energy used. إذا في energy used, then I don't care about how much is here or here. I can move however much I want.
Here doesn't matter.
Doesn't matter.
Okay. Do you guys have any other questions? No, it's okay.
Now I'll start talking about active transport. Okay, we have two ways of transport, either primary or secondary. Primary, easy. Secondary needs some thinking. Okay, what is primary transport? It is the movement of solutes against the concentration gradient. We know that in passive diffusion it moves from where to where? High to low. Here it happens the opposite. Because we're using energy, we can move from low to high. We are using ATP to activate this pump to pump out protons. Okay, so يعني, let's use as an example. Come here. I am using on him energy now. Please come. Please. Outside it's full. Yalla, go out. Full. Outside it's full. Okay, he wants to come back inside. I will not let him. I used energy to kick him out. What is this? ادخل لا ادخل عشان احتاجك عشان المثال الثاني كمل. Make sense? Make sense? If it doesn't make sense, I'll kick someone else out عادي. For secondary active transport, okay, it is when you do what we did now, we remove one, and then you use a co-transport. For example, you exchange. Okay, so next. For example, here we have here sodium potassium pump. You studied this in biochem, so you should know like this already. So ATP binds, sodium binds. It comes when all three bind, it hydrolyzes the ATP. It becomes ATP, leaves phosphate. Now this makes a conformational change. When it makes a conformational change, the sodium leaves, and then for the phosphate to like separate, it needs 2 potassium. When the 2 potassium bind, this leaves, and then it starts the cycle again. Okay, this is easy. You studied it in biochem. The idea here, we have symport and antiport. Symport is when two things, like 2 molecules, they go in together in the same channel. They move in together. Antiport is like this, sodium, potassium, where they wave at each other. They say hi, bye, and هيك passing, and they go to opposite directions. Okay, so one it's antiport is one is in and the other is out, and symport is both in or both out. Okay, so let's give an example for symport. Okay, so now I will use energy, okay, to remove a molecule. This molecule is sodium. Come here, sodium. Come here, sodium. Come here. Outside, what? Outside is what? Outside is what, ikhwan? High concentration, because I'm using energy. Okay?
I'm resisting, but
You are resisting. No. Leave. And you cannot come inside until you bring with me karak. I want 5 karak. Otherwise, you don't come inside. خلاص. ادخل اصبر سيجعل. ادخل. Now this when I tell him come inside and bring with you 5 karak applies to this. The glucose. Sodium, when it comes back inside the cell, what does it do? It brings with it glucose. And what we call this? When 2 come in together? Symport. خلاص, easy. Make sense?
yes.
And this, ولا أقول له ما يحتاج. Vesicular transport. Repeat? I will use it on you. And now for vesicular transport, okay? This is an easy idea, okay? We have 2 ways. The vesicles is like what? A bubble or like a bag, okay, inside a cell. This bag, جلطني الميكروفون. خلاص مش مهم. This bag, okay, it can carry stuff in it. Now, how do you open the bag and close it? You open it, close it, open it, close it. Next. We'll start with endocytosis, which is what? Putting stuff inside this bag. Now, we use this bag for many things. What do you mean? We take stuff from outside, okay? So this is the outside and this is the inside, okay, inside the cell. We can do the first type of endocytosis. You know what's endocytosis, صح? Some of you read the book? Okay, good. Endocytosis, for example, when it comes to phagosomes, you have like pseudopods, which is what? Who knows what pseudopods? What's the literal meaning? Like, what does it translate to? Who said it? From this side, you? صح, fake foot. Pseudopods means fake foot. So these fake feet, when a phagocyte connects to them, the phagocyte will be like a coded pit and it will fall inside. It's like a trap. When it falls inside, it catches it. Okay? We see this normally where, can anyone guess? It's not in the book, بس يعني guess.
What's the name?
صحيح. And where white blood cells, macrophages, stuff like this, they need to catch organisms, big organisms. They do this trick يعني where هذا and then catch it. Okay? And penocytosis, he is thirsty and he wants to drink. يعني there is nothing specific about this. Penocytosis is when it wants to test the extracellular matrix. Okay, to check if it's good or bad. So it just takes any general idea. Yes. ها؟ شنو؟ والله مش سامع.
What is clathrin?
وين شنو كلاثرين؟ ما هو طيب I'm teaching bio not chemistry يا اخي يعني. You should have came at Yasser's lecture, you should have asked him, not mine. For receptor mediated endocytosis, for example, you have, as Ahmed explained, you have receptors. These receptors, they can sense the molecules. Some molecules are very rare. They need them inside them. يعني they are rare in the extracellular matrix and they need them inside. How do they like choose these molecules specifically? لو سمحتوا ترى فرجوا عليه شوي. براع. We used ATP on him to kick him out. Okay? So this is okay. So receptor mediated endocytosis, the receptor feels something and then it comes inside. Now some of you will say, what's the difference between this and this? This uses receptors, this uses podocytes. This is to take something small, like molecules. This to take a whole like what do you call it? Like a prokaryote, يعني a whole like bacteria or something. Okay? Yes. لا ما يحتاج أكلك. لا ما يحتاج أكلك. And this for exocytosis. Should I ask you a question? طيب, let's ask you a question. A molecule was, yes. خلاص okay. What method what method is mostly used by macrophages? And endocytosis. What type of endocytosis is used most by macrophages? صحيح. But because no one raised his hand and I didn't know who said, so no one takes anything. طيب ما هو الشيء سي
معتز
خلاص عطي واحدة ثانية. An exocytosis, it's a simple an exocytosis, it's simple idea, okay? You can imagine it like يعني champagne is حرام يعني, but you can imagine it. إنه this is a vesicle. It has what? A V snare. What does the V represent? The letter V. صح, vesicle. And the V is on what? On the vesicle itself. The T snare. T represents what? Target snare. So when the V snare and the target snare, they bind together so that he can do exocytosis, so remove stuff out of the cell, they will spin, like they will spin together like this, and they will stretch. Make sense? يعني I want to draw it, I don't want to يعني ruin the PowerPoint, okay? So imagine يعني, it's like this, and then it goes like this. Make sense? Anyone didn't understand any part? خلاص okay, next. طيب skip كم سلايد. الحد الثالث أنا بدي أشرح عليها خلاص. Okay. So this will explain how potassium is the key player of the membrane potential. Now normally we have a membrane potential, صح? You took it in biochem? Okay. So cell membrane, the outside is positive, the inside is what? And to keep this membrane potential, what does the cell try to do? First, potassium. Potassium, what does it do? It leaves the cell. Okay, there is leak channels. And when it leaves the cell inside will become more what? More negative. Why? Because potassium is positive. And when you remove positive, this will become more what? So when this becomes more negative, what will happen to the potassium? هو do يعني هو he goes everywhere يعني he doesn't know what to do. He goes here, okay, this looks more negative to me, I will leave. He leaves. He goes here, he goes لا والله, inside is more negative. He comes back inside. And then it continues this, and this is how like it tries to keep the membrane potential at around 90 to 70. Make sense? Easy? ما حد قاعد يسأل ايش السؤال يعني. خلاص. And now back to Ahmed.
So now we'll talk about cell adhesion molecules that are like basically the membrane receptors. The most important molecules cell used to interact with with other environments. They have into two large families, basically the adhesion molecules and the plasma membrane receptors. Okay, we'll talk about the plasma membrane receptors after this one. But basically, the CAM molecules is basically cell adhesion molecule. Okay, from the word adhesion, you can know it means sticking together or like anchor. The word anchoring, anchor themselves to molecule in the extracellular space. Okay, they play a key role in what? In embryonic development and wound repair. For example, let's say we have like a small cut. The cell adhesion molecules, they are like sensors. They help the white blood cells, which like cause the clotting and to like fight the bacteria and the infection, basically. Okay, to come to the place and like give a sensor to the white blood cells to come and protect our body, basically. Okay, it's like a sensor. Okay, and as I said, SOS signals, the one that's like famous in TikTok. It's like calling for help. And also it's mechanical sensors that transmit information about changes in the extracellular matrix. For example, that's like the CAM adhesions, right? And that's the extracellular matrix. If something happened here, if something occurred here, okay, this one is it sends the messages to inside the cell into the cytoplasm. You guys know what I mean? So something happened outside, cell adhesion molecule is responsible to send the message inside the cell. يعني تبعت لها message, oh, there is something dangerous outside. It's like basically, you guys know the alarm that came أيام like too much rain or in Corona, stay in your houses. It's the same thing basically. That's the purpose of CAM adhesion molecule. Okay, you guys get it? Saif اللي بعده. Now we have the receptors, plasma membrane receptors. You have two types: contact signaling, اللي هي both of them touching each other immediately, and chemical signaling, okay, which is like basically sending a ligand or like a specific chemical messenger. So contact signaling, cells come together and touch as like that, okay, signaling cell and the target cell. For example, the bacteria in our body, when it wants to attack something, for example, a bacteria called like the bacteria for tuberculosis, something in the lungs, like infection in the lung, okay. So let's say the bacteria of tuberculosis came to the specific target tissue, which is the lung. It's the specific target tissue. So contact signaling, it's basically the bacteria have a specific receptor for this target cell to infect it. As simple as that. It's just immediately they contact together. Okay. Chemical signaling. Who could tell me, like, give me a brief idea about chemical signaling?
Is the neuromuscular junction as a
Yeah, you can use the of the neurotransmitter and calcium ions. Who else could give me something about the chemical signaling?
Binds to it to initiate
Exactly, it's simple. Also, it's the same as contact signaling. It's just basically sending a ligand or a message. I'll explain it here. Is the process in which a ligand, the chemical messenger, binds to a specific receptor and initiates a response. As مين حكاها? As Mu'tasim said, it's neurotransmitters and hormones, and as she said, it has a specific receptor. Okay? معتز وين؟ حلاوة الشاب. When a ligand binds to a membrane receptor, the receptor structure changes. So basically, when a ligand connects to the receptor, they change the shape. Okay, so it can connect easily. And the cell proteins are altered in some way, like they are changed. The structure of it is changed. Okay, بتتغير. For example, some membrane proteins respond to ligands by becoming activated enzymes. You guys, I think we will take it in biochem. Sometimes they change their shape when they are attached. Okay. While other common in muscle and nerve cells, they are mostly common in nerve cells and muscle cells. يعني, as I said here, in neurotransmitters, basically when I do like this to my leg, it's basically I sent my nervous system sent neurotransmitters to my leg to move it. Okay, that's what causes what the ligand and the channel proteins to the protein channels to open, which is like calcium. Okay, and here as you guys see, that's the ligand, that's the cell, that's the receptor, and it causes a response. تافه. And now you have the plasma membrane receptor. We use the G protein, okay, as like an indirect way to for example, to regulate the molecule. Okay, it acts like as the middleman basically. It's like G protein here. It's like the thing like that causes this to happen. It's basically the most important regulatory like protein. Okay, first of all, what happens? The ligand comes and binds to this receptor. Okay, that's the ligand, that's the chemical messenger, and the receptor on the cell membrane. Okay, also I'm reminding you, what type of protein is a receptor? شكرا. مين حكاها؟ عطوه هذا. Okay, so the ligand binds to the receptor changes the shape and activates. So when the ligand binds to the receptor changes the shape so it can accept the ligand. Okay, يعني every receptor can change its shape according to the ligand or the chemical messenger that's coming. Here you have number 2, the activated receptor. The activated receptor, it binds to the G protein. G protein, which as I said is the middleman, regulatory. Okay, the activated receptor binds to a G protein and activates it. Basically, in this process, what happens? The G protein, it changes its shape and connects to the receptor to take the message. Okay, after it, the G protein goes to the effector protein, which is mostly enzymes. Okay, as you can see here, the activated G also, they use energy here. They produce energy. They turn GTP to GTP. Okay, from GDP to GTP. So there is a form of energy here. Okay. Activated G protein activates or inactivates an effector protein, causing it to change its shape. So as you guys see here, anything that's like, for example, when the G protein connects to the receptor, it changes the shape. When the G protein connects to the effector protein, also changes the shape. Okay. طيب, who could tell me what's like the common messengers for G protein? مفيش غيركم بجاوب انتوا؟
اختار ناس من الورا، اختار من الورا.
ايهم جاوب. شو اسمه؟ What's the most common second messengers? Yes. Yeah. ناس منيح ما شاء الله. Okay. So here we have then the effector protein, which is the enzyme. Activated effector enzyme catalyze the reaction that produce second messenger. So basically, this enzyme, okay, it activates the second messenger. Like this one is already inactive, مو شغالة. Okay, it's not working. This comes and activates it, makes it like active, makes it work. Okay, it makes the second messenger work. And as she said, the most common 2 second messengers you have is the cyclic AMP and the calcium. Okay, so يعني إذا حكوا لكم مثلا what's the most بالاكزام حكوا لكم which one is the most usually cyclic AMP calcium ion على طول انه you guys will know these are the most 2 common ones, and it will also help you analyze the question. It will eliminate stuff. Then you have the active second messengers activate other enzymes or ion channels. These enzymes, they help in opening the ion channels for the calcium, sodium, potassium to pass through. Okay, so cyclic AMP typically activates protein kinase enzyme. Protein kinase enzyme, guys, is basically it takes the phosphorus, you know, like the P, the one in the periodic table. Okay, it takes the ATP, turns it into ADP. It just takes the phosphorus group. Okay, the active second messenger هون. Okay, you have this enzyme, it's activated by the G protein. تمام? Okay, when this enzyme is activated by the G protein, you have a second messenger. The second messenger is usually calcium or cyclic AMP. These are the second messengers, and they're activated by what? By this enzyme, by this like by the effector protein basically. Okay, when هذول a second messenger, they are activated. They activate enzymes that cyclic kinase enzymes. What are cyclic kinase enzymes? They move ATP, like they move a phosphorus group from ATP and turn it into ADP as a form of energy. Okay, so just it's it's an active second messenger. It just helps in the protein kinase enzyme. It just activates them. It's basically enzyme activating an enzyme, enzyme activating an enzyme. Okay, do you guys have any questions? من عيوني الاثنين حاضر. اوكي, طب وين البارت اللي ما فهمت كلها ولا ده افكتر؟ كلها. طيب it's okay. اول شيء I told you احنا عشان the cell to get its information you have a ligand to bind to the receptor. تمام؟ اللايجاند اللي هي هاي الكيميكال ماسنجر to bind to the receptor. الريسبتور هي اللي تستقبل الانفورميشن. It takes the information inside. Okay, it accepts it. تمام؟ After it accepts it, the receptor changes its shape according to the ligand. يعني let's say the ligand is square, the receptor will have a square like shape عشان to accept it basically. Okay, you have the activated receptor, this receptor, it binds to the G protein. With the G protein change the shape to accept this receptor. You know what I mean? كلهم بيغيروا اشكالهم basically. Okay, and then when the receptor is connected او لما تشبك بهاي الجي بروتين GTP is produced. تمام لهسه؟ Okay, then you have the G protein. Okay, when it when the GTP is released, G protein goes and activates هاي الافكتور بروتين اللي هي integral that acts as an enzyme. It acts as an enzyme. هاي الانزيم هي like the key here. لما الجي بروتين بتسوي activate to this one, when it activates this like protein specifically, this protein helps this protein and this enzyme activates this one, بتشغلها. يعني basically the G protein makes this work and this makes the second messenger work. Okay, so the activated effector enzyme catalyze the reaction that produce second messenger. السكند messenger شو هم الاثنين second messengers؟
كالسيوم
وشو كمان؟ cyclic AMP. These two. Okay, when they activate it, second messengers that activate other enzyme or ion channels. Okay, these اللي هم cyclic AMP والcalcium channels, الكالسيوم, they typically activate protein kinase enzyme. يعني active second messengers they activate another enzyme which is the protein kinase enzyme. وهذول protein kinase enzyme شو فائدتهم؟ من بكم شو؟ exact phosphorylation, helps in the phosphorylation. Exactly. Do you have في حدا مش فاهم؟
كثير نشرحها بالاخير مره.
Okay, if there's something like if someone has a question tell me after the thing. Okay, here that's really simple. You guys probably took it in biochem, which is the cell organelles. It's the first of all you have the cytoplasm. It's the cellular region between the nuclear and the plasma membrane. Basically, it contains the nucleus and also the plasma membrane. It's just like the fluid inside, as I just explained. What's its function? It contains like it basically stabilizes the intracellular fluid, like it stabilizes the organelles. It's like يعني بتخليهم يلزقوا بمكانهم. It organizes it basically. It's simple. You have the mitochondria. You guys know all. It's like it has two membranes, inner membrane and like an outer membrane. Okay. And for example, let's tell you they told you a question in the exam that the ATP is not produced enough in the body, or the cells are like they are ATP. They have ATP deficiency. What type of organelle is affected? Mitochondria. معتز. Mitochondria, exactly. It's the powerhouse. It produces the energy. Powerhouse energy, ATP. Basically, that's the form of energy in our body. And we have ribosomes. We'll talk about it shortly in translation. It's basically like the site where proteins are released. هسألكم نفس السؤال. If a cell had deficiency in a protein, what type of organelle is ruined or damaged? Exactly. معتز حلاوة. بس يا أعطيها شو ما بدها. Okay, and you have also the rough endoplasmic reticulum. They are sugar groups attached to proteins within the citrins. Proteins are bound in vessels for transport to the Golgi apparatus. Basically, in the rough ER, it has ribosomes. And there you can also make proteins. There you can like create proteins. And these proteins, they are made by the ribosomes in the rough ER. As simple as that, okay? Then these ribosomes, they go to the Golgi apparatus, which we'll talk about. The Golgi apparatus عبارة عن it packages or modifies. It checks, okay, that protein is good enough for the body, so we can use it. It's just like a package, زي security. We check or know if it's good enough or no. It's like a package or modification, basically. A smooth ER, okay, guys, be careful. You have the rough ER, it produces protein. Okay, smooth ER, lipids and steroid synthesis. For example, let's say the lipid, you have lipid deficiency. Which one is ruined? Smooth ER. You have protein deficiency also, could be a rough endoplasmic, but he won't confuse you. He wants you get like the same like choice. They will get you one. No, smooth, like not in this way. Okay, and as I said, the Golgi here is the one that modifications and it's like packages the protein, okay, to see if it's good enough or no. Peroxisomes, they are sacs that has catalase and oxidase enzymes. These enzymes detoxify a number of toxic substances. For example, let's say you have a toxic substance inside your cell. That toxic substance, our body like takes care of it by which type of organelle? It's the peroxisomes. Okay. And then the lysosome, it's basically digestion. So if you guys said detoxification, peroxisomes. digestion lysosome. Lysosome it basically زي البطن بتاكل بس. Okay. طيب who can answer this question? It's للدارس. It's, to be fair, who studied the whole book can answer it. Yeah, it's B. Why did you choose B? لا أعطيها for the effort يعني. I know it's موجودة. جيبها. بس why؟ هاي مستقصدة. Exactly. Autophagy is basically you're just changing something in the cell without like removing the whole cell. It's just literally the same cell. It's just you're changing like بتسوي شوية تعديلات. You're just doing some fixations to it. Okay. طيب. Now you have the cytoskeleton. Cytoskeleton you have 3 types. You have microfilaments, which is the actin. اللي هي in the muscles. You guys know actin and myosin, صح? So the actin هي microfilaments. Okay. طيب. You guys know what are the function of muscles basically? Contraction and what? بتساعدنا شو of movement, صح? Exactly. Actin is found in muscles, صح? So always remember this when you relate actin to muscles, muscle helps us in movement. So microfilaments helps us what are involved in most cell motility. Cell motility means the movement of the cell, and they give also a stable shape to the cell basically. Okay. So if they told you in the exam the cell had no ability to move because of what? They have a deficiency in what type of cytoskeleton? Microfilaments. Exactly. Simple as that. Because they don't get straightforward questions in the exam. They play with they have to get something and you have to analyze from it. Okay? And now we have the intermediate filaments. Hi. This one, that's what it's found in which type of junction? مين حكى تايت؟ عشان حكته له. تايت؟ desmosomes. Exactly. Good job. Desmosomes لأنه what? It resists tension. هاي اصلا احنا قلنا انه it's made of what the tetramer fibrils. Okay. هاي البروتين مسؤوله. Okay. It's like عن resisting the tension and it's also insoluble, like it's insoluble protein fibers basically. Okay. So it resists the tension. Okay. And it's made of the protein tetramer fibrils. Okay. And it's basically resisting tension. That's it. That's the only function for it. Exactly. Keratin filaments. It's an intermediate filament. As I told you, explain intermediate filaments اللي بتكون بالنص between the membrane for the desmosomes. Okay. Then you guys have the microtubules. Microtubules, it has the subunits of the spherical protein called tubulin. Okay. So the guys might ask you, they might not ask you, oh, what's the function of microfilaments? Might tell you, okay, what's the function of the cytoskeleton that contains actin, which is microfilament? What's the function of the cytoskeleton that contains fibrils? It's the intermediate filaments. What's the function of the cytoskeleton that contains tubulin? Microtubules, exactly. So microtubules, they are dynamic organelles constantly growing from the centrosome. You guys know the centrosomes and the centrioles هذول اللي بيطلعوا microtubules that help in what? PMAT اللي هي mitosis. Exactly, cell division. They determine the overall shape of the cell as the distribution of cell organelles. So if they told you, distributing the cells and arranging them is the microtubules. Determining the motility is what? Is the microfilaments. Resisting the tension is what? Intermediate filaments. Okay, that's basically the 3 functions for them. And now the centrioles and centrosomes basically it's just the things that produce. That's what you all have to, you have to know about. It produces microtubules like the spindle fibers for cell division, for mitosis. That's it. وين شو؟ Now Saif will go on وانا بكمل بعدين. لا لسه. We'll finish now in a bit.
Try to speed up عشان I'll try to speed up شوي يعني I don't want to take much of your time and most of this is يعني at the end is يعني not that hard. إن شاء الله we finish fast. حط يا أخي أساس قاعد أقول fast. المهم we have organelles. These organelles are related to the centrioles and that was explained by my colleague Ahmed. We have cilia, flagella and microvilli. Okay. Cilia and flagella they are like rod-like things that can be moved. That can move. Okay, yeah, صراحة like that. Okay, so cilia. Where did you see cilia before? In tissue, صح? Epithelium. What does it do? It propels mucus, for example. At that case, صح ولا لا؟ flagella, where did you see it? Sperm. For what? To make the sperm يعني move يعني to move the whole head, the whole cell. Okay, so what's the difference between cilia and flagella? هل منكم يجاوب؟ خلاص انت. صحيح. So cilia, the cell is in its place. It moves it moves stuff around it. Flagella moves the whole cell. The cell itself, it moves. That's the main difference. Both of them, the cilia and the flagella, are like the ends of these cilia and flagella. What do you think they're made from? Like the covering itself. صح. This covering is just an extension of the plasma membrane. So they can tell you يعني, oh, he has a problem with the cilia. What part of the cell is damaged? Like part of the external part. What do you say? صح. What else? Cilia and flagella, they have a certain filament that Ahmed talked about. Which one? He didn't.
Motility.
Microtubules. Microtubules. So both of them, these cilia are made from microtubules. They are made in 9+2 formation in basal dyning.
لا motility. You said motility.
لا قلت السيليا والهايم نفسها من شنو مصنوعة.
Okay, sorry.
So made from 9+2 dyneins. Now I would draw it, بس يعني I don't want to ruin the PowerPoint. So let's imagine with me, circle. And inside the circle, these two circles like this, 9 times on the edges of the circle. The picture is in your book. And then in the middle, 2. This is a 9+2. And down more in the centrioles, so if this is all a centriole, here around this part, it will be a triplet, a 9 triplet. So 3 circles next to each other instead of 2. Make sense? It's just a picture. You see it in your book. You just memorize the information. 9+2 in the basal bodies. Okay. And now I will go back to the question I skipped. Oh, well, I will not go back. Wait. Microvilli. I didn't talk about it. I forgot الصراحة. Microvilli is what? We talked about this before also. صح. Increase surface area. Do you have anything here to test on? Paper? ما عندنا. خلاص. Okay. No problem. Surface area for microvilli. I think you understand. The same idea applies. Microvilli just don't move. And what's the difference between microvilli and cilia? Movement, absorption. But what's another difference? A structural difference. Thickness, but another more specific. Not length. يعني more interfilament. يعني what the difference is in filaments. The cilia uses microtubules. Microvilli use actin. Their structure, these like يعني, although this picture is wrong صراحة, but there is no other picture on Google. The microvilli, they increase surface area, صح, like this. Now this, like عمود, okay? In the middle, what is it? In here, in the middle here, it is what? Microtubules, صح? We said it's made from microtubules in 9+2. Here it's made from actin. In this case, which is like you rarely see it, actin is used in structure. And they can bring a question on this. Even though Ahmed said that actin is normally used in movement, here it does not move. It contradicts here, okay? Good? No questions? طيب, okay. Now I will go back. ما حد غش صح؟ Okay. Brush border structure is missing in the new cell. Which of the following is damaged? I explained this. Wrong. Wrong. He said cilia, I told him wrong. Huh? Microfilaments. حطيتها غلط. مش المفروض تكون اكتن؟ صح. The answer is not filament. مشوها يا اخوان مشوها.
اطلب من حدا يشرح السؤال.
Someone explain the question. انت متحمس بزياده صراحه اشرح. طيب. صحيح. فاهم اكثر. Make sense? Did everyone understand? Okay. It's microfilaments. And where do you remember seeing brush border? Now I'm connecting tissues. يا اخي قول سيليا مرة ثانية بطردك. Digestive system. Where do you see it in absorption? Brush border is for absorption. For what? Microvilli. Okay, not cilia. So microfilaments is made from what? And actin makes what? And brush border is, you see how it's connecting? So if this doesn't come in your quiz, it might come in the mid or it might come in the final. Like we got similar questions to these, where they try to connect chapters. Okay? عشان اخوفكم يعني. Okay. Which of the following organelles orchestrate programmed death cell death? هاي المفروض after هذا بس عادي. انت اللي في النص هذاك. صح. صح. We got this question. We got a similar question to this last year, and I didn't explain.
Same question.
We got a similar question, not same. Okay, ما خصني. We got a similar question to this last year. Now, orchestrate, I know. Orchestrate, some people put nucleus. صح? Because nucleus would tell the cell programmed cell death. But who does the action? Is it the nucleus? Does the nucleus يعني bring a knife and يا الله الميكروفون طار like this? No. Who kills it? The lysosome. The lysosome will kill the cell. He orchestrates the death. He is the person that's holding the knife. تمام? Any other questions? خلاص اوكي. And this just explains everything I said. You can. This is just يعني from the book, but I wanted to explain it in my way. Okay. So صراحة I recommend you all read the book. يعني nearly never they go outside the book. يعني like how many questions? يعني we got last year. How many questions did we solve? Like 150, 160 questions. يعني with the mid, with the final, with the quizzes, and nothing was out of the book. For biology at least. Okay. Biochemistry different story. Yep. The nucleus. The nucleus. Everyone knows what a nucleus is, صح? So we'll move through this quickly. Nucleus, it's a nuclear envelope. It's double membrane, and it's double membrane similar to what? The mitochondria. And this I don't know if you mentioned it. صراحة أنا ما كنت مركز كنت قاعد أتفرج الموبايل. Mitochondria has DNA. صح? Everyone knows this. خلاص okay. Now the double membrane, the inner membrane is lined by the nuclear lamina, which is like a scaffolding to keep the structure of the nucleus. And this lamina helps to shape the nucleus and acts as scaffold. And at several points, the nucleus itself, it has pores, يعني like channel proteins that allow the movement of, for example, what? What do we want to leave?
Waste.
mRNA. Waste the nucleus. We want things to leave like mRNA, صح? Because we want it to leave to what? To code for proteins. So that's an example. This is why we needed the pores, for example. Within the nucleus are things called nucleoli, and these nucleoli makes the ribosomal subunits, and like they assemble them. Please silence, please, 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 please. These ribosomal subunits, what are they? There are two. شفتك أول خلاص. صح. Large and small. Simple. صح ولا لا? It makes what? The RNA. At the microscope, there is like chromatin, which appears as fine, and I don't know what. The idea is DNA, and then nucleosome, and then chromatin, chromosome, and then the nucleus. Okay, so you need to know this order. And it's in the book as well. I recommend you see the picture in the book. It's much nicer than this. Okay, we'll talk about histones now. Wait. The composition of a chromatin, which is this part. And let's go back. Chromatin. فهمتوا الكروماتين؟ The chromatin is 30% DNA. 60% histone. يعني there is more histone than DNA. هاي كيف صارت ما أعرف. And 10% RNA. These RNA chains, you'll see them walking around like tRNA, RNA, whatever. They are newly formed or they're the ribosomes or whatever. Now since histone is 60%, يعني لازم نعطيه احترام and talk about it. Histone is these circular things that we saw before. They are like proteins, huh? Packaging, يعني packaging proteins. where they are very special. See this one, this circular thing is a histone. We use this histone to wrap around. يعني you have histone. يعني imagine me and you wrap around me the mic. The mic is the DNA. I am the histone. That's how it works, so that it gets packaged. Easy? Make sense? So let's say like how many do you think it is? يا اخي انت جاوبت ودي حد ما جاوب. خلاص انت جاوب، يلا جاوب، ما في حد. صح 8. So it makes two circles. Okay, so the DNA spins 2 times around 8 histones. And why is histones important? Other than storage or like to like coil it. Does anyone know? Well, no one studied. The other importance of histones, like what else other than keeping it يعني مرتب حلو كتكوت. ما هو مرتب حلو كتكوت. لا انت ما تنحسب. حمدي. مش مركز. خلاص I'll explain. In non-living cells, for example, the presence of a methyl group, you can add a methyl group. You know what's a methyl صح biochemistry؟ كويس الحمد لله. Methyl group on the histone. What makes this? It activates the DNA. يعني, do you understand this? When it activates the DNA, you cannot use it to make proteins. So DNA inactive. Why? Because we methylated the histone. طيب how do we activate it? Acetyl. مين قال acetyl؟ اسطورة. We acetyl the histone so that the DNA becomes unbound, and then we can use it to يعني read it and whatever. We'll talk about this now. Do you have questions? لا we don't. كمل. باقي يمكن 40-50 سلايد. مسخر ما حد يصدق. هاي النيوكليوس اخر جزئيه ما عليك.
لا خلاص كان فهمت.
هاي transcription translation
15 minutes and it's gonna end يعني. So now guys, here will be like the like transcription and translation part. But first of all, you have to know a bit of like structures, so you guys know. So you guys all know, like you are familiar with RNA, صح? You know what's RNA? أخيرا إيش مجهوده؟ أخيرا something from his هذا.
اخيرا جاب سؤال.
Okay, it's ribonucleic acid. At least he tried. Okay, what's different from the RNA and DNA? الله رجاله
اذا جاوب سؤال ثاني بدي اعرف اسمه.
So basically, DNA, you guys know, like genes, they make proteins. They are the original. Like proteins are originated from proteins. Okay, sorry, proteins are originated from genes. Sorry. Okay, so DNA serves as the master blueprint for protein synthesis. Okay, DNA it carries our genes that will do protein synthesis. Each sequence of 3 bases are called a triplet. So also, what's different between DNA and RNA when it comes to like the nucleic bases?
Ribonucleic acid and deoxyribonucleic.
No, I mean nucleic. مش عارف مين Exactly, yeah, uracil and thymine. صح. معتز. أعطيها. Okay, it's uracil and thymine. Just like a brief information so you guys understand. Okay. Okay, so first of all, when DNA is formed to RNA, that's called transcription, and Saif will explain it shortly. Okay, but here I'll just explain some the important stuff that you need. First of all, the messenger RNA is the RNA that's transcripted, basically the DNA that's transcripted, because transcription is DNA to RNA. Okay, translation is RNA to protein. Tamam? So messenger RNA is the RNA that's formed from the DNA, basically. Okay, which is like the blueprint to continue protein synthesis. So as you can see here, messenger RNA carries the message of genetic code from DNA to the ribosome. Okay, يعني when we finish transcription, messenger RNA goes to the ribosome, so we can do translation in the ribosomes. Okay, and you have the ribosal RNA. Ribosal RNA is simple. They make ribosomes. Okay, and transfer RNA. It's mostly important in translation. You guys know translation is from RNA to protein. Okay, so transfer RNA, it transfers the amino acids, okay, to the like mRNA codon, basically to the mRNA, and the mRNA has 3 like letters. Every 3 letters is called what? A codon. معتز. Okay, it's called a codon. تمام. These you don't have to know them. They told me Abu Bakr, like the doctor canceled them. The micro RNAs and the small interfering RNAs. They told me it's cancelled. No, these ones are not coming. With the Dr. Osman? But no, في أبو بكر section they're saying it's cancelled. خلاص. Huh? No, these two, just not them in general. In a micro, like they are basically they are against the mRNA. Okay, and small guys. And the small interfering RNA, it's basically the RNA that helps in viral replication. But just take a brief idea about them. As I know from information, it's cancelled. Like you don't have to know them. Okay. Also, just for an mRNA, okay, coding regions which will be proteins are called exons. And the ones that are not like coded, they are introns. Introns like useless information. You don't need them. But exon is the important one, so you can have protein synthesis. Okay. So here, you guys are familiar with this, of course. Like people who take AP, you are familiar with mitosis. Actually, anyone. Okay. First of all, as you guys know, interphase. It has like it's the phase of replicating DNA. To check the chromosomes, G1 and G2. Okay, so first of all, you have G1, it checks the DNA. Then you have the synthesis, the S phase, which basically DNA is replicating. It's like it's synthesizing itself again. And you have G2, it checks if like the replication is right or no. Okay, that's the interphase basically. Then you have here the prophase. Prophase is the first step of mitosis. The chromatin coils and condenses, forming bar-like chromosomes. So the chromosomes start like aligning together, like forms, and it has the information. Each duplicated chromosome consists of 2 identical threads. Basically, the chromosome is basically a chromatid. It's like these chromatids. We call them what? Sister chromatids, which is a chromosome. Okay. They are called sister chromatids, and they are held together by centromere. Centromere is this. You guys see this small part here? That's the centromere. That's the centromere. That's like in the middle of the sister chromatids. Okay. And as the chromosomes appear, the nuclei disappear, and 2 centrosomes separate from one another. You guys see these 2? They separate. ليش? Because later they wanna send microtubules, spindle fibers, to attach to the chromosomes so they can go through metaphase. Okay. And basically here it's just like the chromosome starts to condense more, starts to show more. Then in the late prophase, all you have to know is like the microtubules which are released from the centrosomes. Okay, the microtubules start attached to the kinetochore. Kinetochore, guys, is the protein that's attached to the centromere. That protein helps attach the spindle fibers to the middle of the chromosome. Okay, فإذا حكى لكم, let's say there is a problem with the kinetochore. What would happen? The spindle fiber cannot what? Exactly, as simple as that. Okay, and also the nuclear envelope. You guys see this thingy here? This is the nuclear envelope. So they told you what happens in late prophase, for example, the nuclear envelope disappears, so the chromosomes can align in the cells. They can يمشوا على راحتهم basically and align as in the equator and metaphase. Okay. That's it. And then like late prophase, the spindle fibers attached to kinetochore, and then what do you have? You have metaphase. Okay, guys, can you tell me what characteristics about metaphase? The line in the middle of the equator. مجريش أعطيها لو سمحت. Yes, tell me.
Or just from there?
No, it comes from there. تطلع من هناك. Okay, بتطلع من الcentrioles والcentrosomes. Okay, it comes from this like it's called like an iron triplet. You will read it in the book. Maybe you read it or no, I don't know. But like it's a triplet and it releases spindle fibers. I explained in the previous chapter slides. Saif also explained. طيب, you have metaphase. Metaphase, it's the most like part of the cell cycle, I mean of the mitotic division. You can see the chromosomes in. So if they told you a scientist wanted to see the chromosomes in a proper way, or like in a really like, let's say a full HD, which type of mitotic division like phase? Metaphase, because they are lined in the equator and you can see them like properly. Okay, and be careful guys, they might get you what chromosomes are condensed the most in metaphase. Condensed يعني they are showing the most. Okay, that basically it's just they align on the equator. Tamam. Anaphase is basically you're just separating the sisters away from each other. You're separating the sister chromatids, everyone goes to the opposite pole. Okay, that's it. That's all you have to know. It's also the slowest, and the kinetochore of the microtubules they detach and they become smaller. ليش؟ لأنه that's the kinetochore, صح؟ It's bigger, صح? When you separate it, it becomes small. It's like divided into two pieces. Okay, that's it. And here you have the telophase. Telophase is basically colors. The cytoplasm starts like like the two cells like start separating. It becomes to like خلاص, it will be in the spectrum of becoming what? Two cells. And as you can see here, you have the contractile ring that will then the cytokinesis will help it into dividing into two cells exactly. Telophase is just what? The nuclear envelope. Also, it's basically you're doing prophase but in reverse. Like as I talk in prophase, it's literally like it's going back to this again. Basically, it's just the reverse. Okay, it's just the nuclear envelope is is occurring again. It's forming again, and the chromosomes also align and they are attached together, and then they replicate to divide. يعني هذول هسه they one chromosome, صح؟ هذول they will replicate, then go another mitosis, and that's it. Then you have the cytokinesis. Cytokinesis, from the word cyto, it's the definition of the cytoplasm. Exactly. So you have these. It's telophase, صح? Then the contractile rings help in cytokinesis. Okay, they help them what? Separate these two cells عشان they become two separate cells وكل واحدة كمان تسوي mitosis again and goes through the same cycle again. Is there any questions to this? Yeah, and this contractile ring is actin and myosin. Like any it's like a contractile. Anything that's really contractile is what actin and myosin. It's like a muscle. Okay, and also just one thing, there is also called cyclin kinases. Okay, these المهم cyclin dependent kinase or cyclins. Okay, they just regulate the cell cycle. That's all you have to know. This might get you this question. It's the enzymes that regulate the cell cycle. Cyclin, they are called cyclin or cyclin dependent kinases. هذول هم اللي بيعملوا regulation to the cell cycle. بس. Okay, just note general information. And now after this we'll go to transcription and translation. We'll just explain the figures. So who can answer this question? In which phase of mitosis does kinetochore shorten and non-kinetochore grows? شو؟ لا خلينا نسمع منه أمانة. مشان الله ركز. Focus please, for God's sake. For God's sake, focus. Like read the question properly, please. صح. Why is it C? Who could explain to me why is it C? Exactly, they separate. Exactly. Okay. يلا سيف. طيب. Why does the nucleolus disappear during mitosis?
This question?
No.
ما جاب
It's not A.
Similar question.
Okay. والله يعني هو شيء ما هي خلاص حق. Why B? Why B? No. Yeah, like, but why would you guys say B? Exactly. You already have the DNA and it's already synthesizing in the G1 and the G2 phase. Like you don't need to have a nucleolus again. Okay, that's it. You don't have it during mitosis. It's already in G1 and G2. like هاي الأشياء بتصير. Okay.
I just want to add that this question, although it may look easy, in our similar question that came in the last year, It was much harder because of the choices. I don't really remember the choices. بس يعني truly focus on the differences between G and S and stuff like this. I'll skip. I'll do fast. Don't worry guys. خلاص عارف مليتوا مني يعني مش لهالدرجة. أنا مليت من نفسي. فنخلص على السريع. Okay, please silence. 5 minutes I'll finish. DNA. What is DNA? DNA is a music recording. We use DNA to make RNA. For what? To make a protein. Easy or not easy? Gene is a segment of that DNA. Simple definitions. Information that, so what does it use? CG80 nucleotides. Easy, it's the language. CG80, it goes in threes. Everyone knows this information? Easy? It goes in a sequence of triplets. Exons is the part that will be coded. Introns is the part you will cut out with splash zones, an enzyme that could come in your exam. So when they say, okay, what removes introns? يعني تاني قلت هذا ما جبتوها بزعل. RNA protein synthesis, like we said. RNA, what's the difference between RNA and DNA? We talked about this. RNA is oxygenated and it has U instead of T. This is a really important difference. When they tell you to translate from DNA to RNA or from RNA to DNA, they might give you a sequence and tell you to translate it. So you need to know this difference, U and T. Okay? There are 3 types of RNA. mRNA, which is the long chain that will be developed into a protein. We know mRNA, صح? Easy. Which resembles half of the DNA. يلا عشان rRNA, we talked about rRNA. It's the ribosome with the small and large subunits that will come, bind to the DNA, slide on it, and you'll see a picture on it, and make the thing. tRNA is the L-shaped that brings amino acids and sticks them on the in the correct chain, which Ahmed will explain. But first, questions. يلا على السريع, questions. Messenger RNA is needed to determine which of the following. نشوف مين الشاطرين. ما جاوبتي. ما بعرف صراحة. Is it B? صح, it's B. طيب why B؟ ما يعني بدي حد يشرح لي, أنا ما بعرف. طيب اوكي, في هيك ناس يعني. What enzyme binds to the tRNA? طيب خليني اخلص السؤال بعدين ارفع ايدكم. What enzyme binds to the tRNA to the amino acid? مين ما جاوبتي؟ كلكم جاوبتوا يعني, ما حد منكم جاوب. مش انت, هي. اي؟ لا يعني اهلي يجاوبون, انت تجاوبين. ليش؟ ما خلاص صير. صح. يعني هي حرفيا بس هيك. المهم, transcription, my part, very fast. Initiation, RNA comes, he sees DNA, he sees the promoter region, it's unwinded, it looks nice, he is hungry, boom, he sticks. When he sticks, he will start moving, he will slide, wee, like this. When he starts moving, what will happen? He will code for mRNA, he will make mRNA. We have 2 strands, we have the coding strand and the template strand. Does anyone know why we call it coding and template? What's the difference? يلا انت.
Coding codes for the sequence of the amino acid. Templates works يعني هي اللي يحصل منها
كلام صحيح. So this coding strand is similar to what? Is similar to the mRNA. The difference is in what? صحيح. So this top one is nearly identical to the one that's being made. That's why we call it the coding strand, because it's the same code. يعني هيك تحفظها يعني. template strand is the one that's opposite to it. Complementary. So this complementary strand is the template that you build on. يعني كأنه اسم على مسمى, سبحان الله. After they're done, then you'll go to Ahmed's part. يلا Ahmed's part. اقول سبايس كنسل خلاص يدرسون بالبيت. المفروض مش بالتسجيل, يدرسون بالبيت. اوكي.
خلاص, that's 2 slides. Okay, first of all, now we will talk about translation, which is the step after what? Transcription. Okay, it's basically the RNA is turned into what? Proteins. Okay, it's like into the amino acid sequence. Okay, for each triplet, for example, I told you in the mRNA you have a codon, which is like, for example, AUG, GGGG, AGG, anything, any three letters حطوهم مع بعض هتطلع معاكم amino acid. Okay, and the tRNA, it has the anticodon. These anticodon, they come and like interact with the codon, okay, to form what? To make the amino acid chain or the polypeptide chain, basically. Okay, so translation, it involves what? The three types of RNAs: mRNA, tRNA, and rRNAs. Okay, and we explained them. mRNA is the messenger one that has the genetic information. tRNA is the transfer RNA that takes the amino acid and transfers it into the codon. rRNAs are the ones that produce ribosomes. The amino acid, first of all, the amino acid is picked up from the pool by the what? By the tRNA. Okay. At the other end is the anticodon. As I told you guys, it's like the anticodon that would connect to the mRNA codon. Okay, and it's carried by a specific enzyme. What's this enzyme? Exactly, amino acid tRNA synthetase. It basically takes the amino acid from the pool and then puts it on the codon. That's it. And then the codon بيكون عندها زي complementary. Okay, يعني what? يعني for example, A is with U and G is with C. Okay. This part they told me also it's not with you guys. Abu Bakr said it's not included, so you guys don't have to know it. Okay, مش عارف شو. لا لا. طيب. Okay. Listen. So guys, as I explained here, our issue have three like steps: initiation, elongation, termination. Initiation يعني بتبدأ. Okay, initiating. Okay, أول issue you have the ribosome. It has a small subunit and a large subunit. Okay, so the mRNA it leaves the nucleus and goes to the cytoplasm, then goes to a ribosome. Okay, a small ribosome subunit. The initiator tRNA carrying the amino acid methionine. So you guys also have start codons and ending codons, basically. Okay, or stop codon. Yes, you have the start codon. It's known AUG. أو هقول لكم what type of amino acid? No, it's this methionine. Okay, it's the amino acid that discussed for the start codon. It's what AUG. Just know them. Okay, and a large subunit. So basically, you have also three sites in the ribosome. You have A site, B site, E site. E site is basically like exit. P, exactly. A, amino acid, exactly. Okay, safe بعدين. In elongation, you have three steps basically in elongation. First of all, amino acids are added one at a time via the growing peptide. You have our issue what the codon recognition. First of all, you already have like here the growing peptide which is in the peptide like site. Okay, so then when a new tRNA picks up an amino acid, it has the anticodon. Then this is GAU. Okay, it will connect to what? This codon. This codon is an amino acid. Then in the peptide bond formation, this tRNA will go into this side. Okay, like then it will go to like هتغير كل شوي يعني واحدة بتدخل بعدين هي بتدخل مكانها. You know what I mean? يعني the anticodon of an incoming tRNA binds with the complementary mRNA codon. Okay, here the site of A ribosomes. تمام? You already here have like a peptide chain. It comes in the A site. Okay, in the amino acid site. Then this polypeptide chain it goes to the A site. It connects to the tRNA in the A site. Then this one, it's kicked by the exit. إنه خلاص it's kicking her. And P is like for parking. It stops there. Okay, the A goes here and it stops at the P. And then it goes on. واحدة بتنزل على A. Okay, like a tRNA بتنزل على A. Then the P gives its polypeptide. Then this goes and you guys know that like خلاص and so on until what it reaches a specific codon, which are the stop codons. You guys know them: UGA or UGA, UAA, and UAG. When they spot these, means خلاص it's the end of translation. Okay. It's when a stop codon UGA, UAA, or UAG arrives at the A site, when it arrives here, it releases a factor that stops translation. You guys have any questions about it? Are you sure? That's like our session for today. We just have like extra questions. Which of the following enzymes bind the tRNA to the amino acids? amino acid synthesis.
Which of the following amino acids is the first moiety in the polypeptide chain? This is a similar question with the word moiety. Subhanallah. مين ما جاوب؟
خلاص we already know. You know the answer. I know.
شرط اسود ابيض ما في غيرك يا اخي قاعد طلع واحد. زينين.
صح بثايرين. Yes.
صحيح.
You guys know what the word moiety is أصلا؟ طب اشرح يلا يا فهيم. اشرح يلا. اشرح.
المهم moiety is like what do you call it يعني ما هو يعني a part.
The first part of translation basically, which is the start codon. Methionine, AUG.
Yeah, the first part of the moiety, the first part of the wording. They will try to bring weird words like this, and you saw some of this in quiz 1. Expect more from quiz 2, especially because they have more يعني, vocabulary in the book. يعني bigger chapter, longer chapter, so they have more flexibility. AUG important makes methanamine. We got a similar question. AUG is methanamine. Don't ignore it, please. And what's not the stop codon?
Hi, hello, After Effects Saif. Like you're seeing, the audio got cut out, so I'm gonna say, we said what codes, like what's the stop, like what's the thing that gets added? It's water or hydrolysis. Like everyone said it's nothing, it's hydrolysis or water or whatever it is. Might come, might not. Study the enzymes from the book. Enzymes important, very important. They might come, they might not, but yeah, better safe than sorry, because some doctors like they really like focused on them. Also, what do you call it? Read the book a few times, please read the book. The book is useful. What else? Book is useful, read it 3 times at least. Thanks to Ahmed Emam for coming today and giving us this amazing lecture. Amazing person. Saif is the one talking. Thanks to everyone who attended, and inshallah you all benefit from this video, whoever is watching. Good luck in your biocell quiz. Inshallah you all will do amazingly. And good luck.